My computer took a little bit longer to get fired up here than usual. Patrick, Isabella, David, and Faith. All the good people are here today. Okay, let me just get my my acting gear here. And okay, quick check-in. Can everyone hear me? Patrick?
Yes.
David, I don't hear you. Patrick, I can't hear
You can't.
Yep, I can hear you.
Can you put your camera on? Give me a thumbs up if you can hear me. Okay, good. Excellent. All right, so quick announcement before we move on. I finished grading the exam on Monday, but I still have 3 people who had computer problems or whatnot and couldn't do it. So I will go over the test when we get back after break, because I don't want to go over it now and then have them get a preview. So I'll do that. What else did I want to tell you? Can't remember. I guess that's about it. So this is our last class before spring break, and so I'm going to do most of chapter. Oh, here we go. We can hear you, but we can't see your screen share. Oh yeah, I haven't started sharing my screen yet. I'll do that in a second. So I'm going to get out of this now. All right. Damn you, Teams. Oh God, how do we get back to where I was?
In the window, yep, in that window you just went over that has Patrick's bubble in it. I think that, yeah, that one.
Okay, we're back. All right, I'm going to share my screen, and then Patrick, I'll check in to see if we're rocking and rolling here. Okay. Well, I can't hear you, so how am I gonna know? All right. So can you see my PowerPoint? Patrick, can you see your face there for a second? Good. Okay, you see the PowerPoint. Good. All right, so we can go ahead and get going. So yeah, you've got your, the other thing about your exam is the class average is lower than the last one, of course, because you didn't have the game to play that gives you bonus points. So I gave everyone 8 bonus points. So you can add that on your score if you want. I just put it into the bonus points category because that's kind of how I've been keeping track of it. So whatever score you got in the exam, add 8 points to that, and that's your final score for that exam. And that's about it. So let's dive right in here. So quickly going over mitosis, which I hate going over mitosis because it's so bio 101. But anyway, here's our cell cycle. This is what we're going to be dealing with. We talked about this G2 to M last time, and we talked about this metaphase to anaphase transition last time. This time we're going to be focusing on the R, which is a bit more complicated than the G2 to M. And complicated means exciting and interesting, right? Am I right? That's what a cell biologist say anyway. Okay, so prophase, 3 things going on in prophase. That's what you really need to know here. There's 3 things going on. Your nucleus is dissolving because of or nuclear membrane is because of the nuclear lamins being phosphorylated. The chromosomes are condensing because of the phosphorylation of condensin. And the interphase mitotic spindle is being reformed into this, or interphase cytoskeleton is being reformed into the mitotic spindle because we're activating non-motor maps. So all those things are being activated by our maturation promoting factor, which is cyclin B and CDC2. And later we saw those targets being phosphorylated, which is, I love when you can make that connection. So it's not just magic happening on the screen.
What did you say was phosphorylated to make the chromatins condense?
So the question was, what was phosphorylated for the chromatin? That is the protein condensin, which is great. I love a protein that tells you what it's going to do. Okay, so metaphase. Remember, we've got some active pulling of the chromosomes. Once the microtubules attach to that kinetochore, the chromosomes are being pulled in either direction, and that makes them line up here at the center. And the cell pauses there to make sure everything's connected to the mitotic spindle. And then we go through to anaphase, and we've got two things going on here. Usually they say anaphase A is when the sister chromatids separate and start moving to the pole. And then anaphase B is when the cell starts to stretch. So we've got those two things going on. And then telophase, I like to say, is the opposite of prophase. Everything that happens in prophase is going to reverse here in telophase. So we're going to get the nuclear membrane to reform. The chromosomes are decondensing, and the cytoskeleton is going back to its interphase form. Okay, so then we talk about the mitotic spindle and the different aspects of it. Oh, wait, for cytokinesis, which is actin and myosin based. Just had a crazy thought. Wouldn't it be unfair if I added drugs to the next exam that we talked about in the first exam? So would mitosis be stopped by phalloidin or colchicine? Aha. That's not fair at all. Phalloidin affects microfilaments, so it would bother this cleavage, but it wouldn't bother the mitotic spindle. But colchicine bothers yeah, I'm not going to ask a question like that. That's mean. That's just mean. I'm not that mean. I'm mean, but I'm not that mean. So we've got three different types of microtubules that are important here based on their, of course, they're all the same. They're all microtubules, but just based on their function. First, there's the astral, and the astral, starting at prophase, when the microtubules, when the centrosomes are together, astral microtubules shoot out to the poles and then drag the thing to the poles. So that's one of the things astrals are doing, separating those asters and putting them at the poles. And the other thing is just basically anchoring this to the pole, so that when the microtubule starts pulling on the chromosome, this is anchored and the chromosome comes towards it. The other one then is your kinetochore microtubule, which is being yanked on from both sides. So the chromosome is pulling itself down while depolymerizing. And then you've got another motor here that's pulling the microtubule in while depolymerizing. So both those activities are happening there. And then you've got your polar microtubules that are, they're actually polymerizing as the motor is walking on it, which is forcing those asters apart and stretching the cell. So, and another question I like to ask sometimes is, what motor is here versus here? And that all comes down to kinesin and dynein. Kinesin goes towards the positive end, and dynein goes towards the negative end. So this motor is moving towards the negative end, so it's a dynein motor. This is moving towards the positive end, so it's a kinesin motor. And here, the motor is walking towards the positive end, so that's kinesin. And here again, walking towards the negative end, so that's dynein. And again, I like doing that because it ties together the old stuff with the new stuff and kind of, making it all one piece instead of separate projects. One of the things that drives me nuts about my undergrads is, like, after the test is over, they take their head and shake it like an Etch-a-Sketch, and everything that happened before is gone. And we're starting fresh. You guys are grad students. I want you to hold on to this material. It may become necessary sometime. Okay, so here we are stretching the cell with our polar microtubules. And you can also see the kinetochore microtubules dragging the chromosomes. And there's the kinetochore. Okay, so then we talk about this, the G2 to M transition. And that is based on, here's some of the history stuff, not that important, based on cyclins and CDKs. So the CDK is the kinase, cyclin-dependent kinase, and that's going to phosphorylate targets and activate things. The cyclin is the control molecule that will turn the CDK on. And we've got 4 main CDKs. In the last class, we talked about CDK1, which is also called CDC2. I tend to use them interchangeably to get you used to the fact that you can use either one. So keep that in mind. And then today we'll talk about these, CDK2 and CDKs 4 and 6. 4 and 6 will get us up to the restriction point, and CDK2 will get us through it. And then cyclins, then we've got 4 we're mainly concerned with. You can tell there must be cyclin C, right? I don't know where it comes into play here. So these are the 4 we need to know. Cyclin D is key to the restriction point, getting through the restriction point, as is cyclin E. We'll talk about these 2 today. Cyclin A gets us through S and into G2, and then cyclin B is the one that gets us through the G2 to M. So let's talk about that G2 to M. Okay, so originally I had this slide just kind of generically about cyclins and CDKs, but now I've made it a little more specific about our G2 checkpoint. So in the G2 checkpoint, cyclin is building up slowly until it reaches some threshold. And when it reaches that threshold, it binds to CDK2, or it becomes active with CDK or CDK1. And the active complex of cyclin B and CDK1 is called maturation promoting factor, MPF. So MPF then, is activity is going to rise really sharply. You'll rapidly go into mitosis, and then cyclin B is rapidly degraded, and that shuts off MPF. And then cyclin B then will build up again. Okay, so let's see. So bottom line, cyclin B, CDK2 is what's getting you through G2 into M. And we'll talk about that in a second. But what I find most interesting about this transition is you've got a rapid transition into mitosis. Cyclin B may be building up slowly, but MPF activity shoots up sharply. And we'll see this again too with the passage through the restriction point we've got, because what makes things have a threshold and activate quickly is a positive feedback loop. You see this all over the place in biology. When there's a threshold effect, there's some kind of positive feedback loop that makes it happen. So what's happening here is when you've got cyclin B and CDK2 bound together, they're not active this way. It needs to be phosphorylated to be active. But before we activate it with phosphorylation, we're going to inhibit it with phosphorylation. One of those kinases is WEE1. There's another one mentioned in your book. I forget the name of it. But now we've taken an inactive complex and inactivated it. So it's very inactive, super inactive. And then we're going to give it that activating phosphorylation, the yellow in here. And it's still inactive because it has these inactivating phosphorylations on it. And then this is the positive feedback loop. CDC25C is a phosphatase, and its activity is regulated by phosphorylation. So it's got a very slow activity originally, and that makes this our rate determining step. I know you're taking biochemistry now, you know what an RDS is. So this is our RDS. And when you've got a rate determining step, that means everything builds up to that point. So all this keeps happening, and you're getting more and more of this building up until you've got a lot of this, and a little bit of it leaks through, and it comes back and stimulates the phosphatase, which means more of it can get through, more of it stimulates that phosphatase, and you have that positive feedback loop. So your MPF activity goes from almost nothing to peaking really quickly. So you've got this slow rise of cyclin B and a steep rise of its activity. And then, of course, it's going to shut off really quickly and come back down really quickly. We'll talk about that today as well. Oh, we talked about it Monday, I should say. And the reason for that steep drop off is because we're going to ubiquitinate and degrade cyclin B. So I've been talking about ubiquitination all semester. Here's the bottom line. You've got two kind of general ubiquitin ligases. Well, let's start back. Ubiquitin is a small protein, and it is just kind of everywhere. It's what we call ubiquitin. And it's picked up by E1 ubiquitin ligase, which passes to E2, and then E2 becomes kind of like a cofactor in E3. Now, there are a lot of different E3s, and each of them is specific for a different substrate. The one we'll talk about here is anaphase promoting complex. So when APC is active, it's going to grab cyclin B and then using E2, put a chain of ubiquitin on it. And that ubiquitin chain then targets it to go over to the proteasome, which strips the ubiquitin off to be recycled and breaks the protein down into peptides. So that's our ubiquitin pathway. And again, we've been talking about this quite a bit. Here we are, living the dream here. Here we are, living the dream, watching that E3 ligase. Okay.
And this is why it degrades so quickly.
Right, this is why it degrades so quickly, because it's targeted to get rid of it really quickly. And we'll see that all the way through the cycle. Most of these cyclins are going to be targeted. Okay, so to get back to our G2 to M transition here, we've got, once we have active MPF, we get phosphorylation of the nuclear lamins, which gives us nuclear envelope breakdown, phosphorylation of condensin to cause chromosome condensation, and then activation of the non-motor maps to rearrange the mitotic spindle. So that's prophase right there. And then the other thing that's going on is we're going to activate APC. So MPF is going to phosphorylate APC, and it kind of primes it. It's not what's going to activate it, but it's going to allow it to be activated. And that APC, once it is active, will break down cyclin B. But mainly what APC is doing is getting us from metaphase to anaphase. And the key to that transition is making sure that all the sister chromatids are attached to the mitotic spindle. Because if they're not, you get aneuploidy. You get two sister chromatids going in one direction, none going in the other, and both cells end up dying because you don't have the right number of chromosomes. And of course, if it's during meiosis, that's how we get non-disjunction, and the process is pretty similar to what we see in mitosis. So similar kind of a thing. So we have to make sure we're attached to our kinetochores. So here's how that's done. So what's stopping us at the M phase is the cohesins, which are binding sister chromatids together. Like I said, there's this tension in the chromosome, and it won't break apart because the cohesins are there. But that tension is bringing you to the metaphase plate. What's going to allow that to release is the enzyme separase, which is a protease that'll break down cohesins. But securase is stopping separase from working by binding to it. The trigger then that caused the transition is the MAD and BUB proteins here. MAD and BUB are holding CDC20 on the kinetochore and until microtubules attach. So unattached, they're holding CDC20. Once they attach, they release CDC20. CDC20 then becomes part of the APC complex. And this is our ubiquitin ligase that's going to cause a breakdown of a lot of different proteins, but what we're mostly interested here is cyclin B. And this is kind of how that process goes. So MAD and BUB holding CDC20 here. Once you're attached to the spindle, CDC20 gets released, binds to APC, and now this is active. And that active APC is going to ubiquitinate securin, gets broken down, and releasing separate. So I get these flashes because, I'm a genius, we're all geniuses. That's why we're here. Just got this flash, a possible essay question, which should be a softball. Name a protein whose concentration is controlled by ubiquitin and what it does and why ubiquitin is there. That's kind of a general question because it gives you a lot of different targets to talk about. So anyway, did I say that out loud or was I thinking that? I don't know. But anyway, once we've activated APC, we're going to break down securin, allowing anaphase to start. And we're also going to break down cyclin B, allowing mitosis to stop. Right? Because if this didn't stop, then the cell would just stay, you wouldn't get cytokinesis and the cell would just stay kind of stupidly in telophase. Okay. So that's what we did last time.
And that last figure we were in, that the depiction of like the red and yellow. So that's the same depiction when we were in the ubiquitin figure that had the E1 and the substrate with the ubiquitin chain. That
right. This would be that red and yellow balls.
Okay, cool.
That's your E3.
Yeah.
Okay. All right. So what we want to focus on today is this. And this transition's more complicated than this one. And the key thing to keep in mind about this one is it's all tied directly to growth factors. Okay, if you've got cells in culture, you have to grow them. You have to keep them alive with serum. If you don't have serum in there, they're going to die. They're going to go quiescent and eventually die. So serum, as you probably know, is like you take blood and you coagulate it. So you're basically activating all the platelets. They release platelet-derived growth factor. You spin that down to get rid of the cells, and what's left is serum. So you've got active growth factors in serum. So to keep cells in culture alive, you have to keep giving them serum because that's going to stimulate this cell cycle, because this restriction point is all dependent on growth factor activity. So anyway, here's how we can see that. So if you remember, our receptor tyrosine kinase pathway, RTKs cross-phosphorylate, they bring up GRAB2 SOS, that activates RAS, RAS activates RAF, RAF activates MEK, MEK activates MAP kinase. And then MAP kinase then goes into the nucleus and phosphorylates some transcription factors. This is Jun, and this is ETS. Jun binds to phos. They together become a transcription factor that we call AP1. And ETS and Jun cause the transcription of MYC. MYC's really important, and they also cause more Jun and phos to be made. So these are what we're going to call immediate early genes. So as soon as you stimulate with serum, you get IEGs being made. These IEGs, a lot of them are going to be transcription factors. So the RNA goes out of the nucleus, it gets translated, and then the transcription factors get transported back into the nucleus. And then we get transcription of our delayed early genes, DEGs. So these are immediate early genes, and these are the delayed early genes. So we get a cascade of gene activation here. Over here we're talking about a cascade of kinase activation. This is a cascade of gene activation, and you can see that in this this figure here. Now I want to tell you right now, I'm switching to a different textbook. The textbook I'm using here, our textbook doesn't cover this well at all. So what I'm using here is Weinberg, The Biology of Cancer. There's a copy of this book over in the Howe Library. I'm going to have them bring it over to our library here, and it will be on reserve there. Mainly what I'm doing here is going from chapter 8, which is about control of cell division. So if you want to read it in a textbook, I mean, I'm sure I'm explaining it fabulously because that's what I do. But it's always good to read about it too. So This will be on hold in the Dana library here. And then I'm gonna have them order the new version, which I haven't seen myself, but I'm sure it's good. So anyway, this is where I got this figure from, and a lot of the figures that are proceeding from here. So anyway, we talked about cells needing serum. So the way they do this experiment is they starve them for serum. The nice thing about that is, you know, if you've got a plate full of cells in serum, they're all dividing on their own time schedule. You know, cell cycle clock is moving, and you need to synchronize them if you want to. You can't, at least at this point, this is probably in the 1980s, you couldn't see individual genes being turned on and off in one cell. That's not possible. It wasn't possible then. Now you can. So back then, it's like we want to see what genes are being turned on by growth factors. So they serum starve these cells, so they're all at G1. They're all at that restriction point, and then they hit them with serum. We're going to see what genes get turned on. So what you see here's control, right? Serum starved. The blue are genes that are off or at low transcription level anyway. Red are the ones that are being actively transcribed. So as soon as you hit them with serum, 30 minutes later, you get all these genes being turned on. So these are your immediate early genes. And 30 minutes isn't long enough to do transcription and translation. So everything that's going on here is based on the protein machinery that's there when the growth factor hits. So basically, all of these proteins are already there, right? And they're just waiting for the activity for the growth factor. Once they do, immediate early genes get turned on. And you can tell that because if you hit these cells with cyclohexamide, cyclohexamide blocks protein translation. So you hit these cells with cyclohexamide, you still see the activation of immediate early genes. What you don't see is the delayed genes coming on, because these immediate early genes have to be transcribed and then translated. They come back in the nucleus, and now we get delayed genes turned on here and here. And these delayed genes, a lot of these are also transcription factors. So once they get translated and transcribed, an hour later, you start getting other genes being turned on. So you get this cascade of gene transcription. These genes here, the one at 120, these are going to be proteins for S phase, right? All the enzymes you need to replicate the genome, they're going to get turned on last. So you've got your immediate early genes, which are transcription factors, your delayed genes, which are going to be things like cyclins and CDKs to control that restriction point. One of them being cyclin D. That's going to be the key nexus point for us here. And then you get genes for S phase. Question.
Just to put this time scale into perspective, how long does the cell cycle take overall? So how long is this checkpoint
Good
take?
question. So he's like, how long is the whole cell cycle? When a cell is in the cell cycle, you know, it varies based on cell species and all that, but you want to assume about 24 hours. Okay. So, and mitosis itself is 30 minutes. So 24-hour schedule. Yeah. Which when you think about prokaryotes, you know, eukaryote, E. coli can replicate in 15 minutes, you know,
Yeah.
so a lot slower.
And your cells are spending probably 16 or 18 hours a day in one of the G phases.
That's right. It's 23.5 minutes. It's either in G1S or G2. Yep. Most of the time you're there. Okay. So, all right. So here we are looking at it. Same thing in a more graphic way. So our growth factor hits the RTK, cross-phosphorylation, activation of the kinases. Here's our MAP kinase here, moves into the nucleus, phosphorylates transcription factors, Jun and ETS. You get transcription of immediate early genes, Jun, Phos, Myc, and a bunch of other things. Jun, Phos, and Myc are transcription factors. These mRNAs go out into the cytoplasm. They get translated on the polyribosomes, they get transported back in, and now we get delayed early genes being activated. Cyclin D, cyclin E, and this bad boy, E2F. E2F is important. So E2F is a transcription factor, and that's the one that's going to turn on those 120-minute genes, right? The ones that actually, the genes that make the enzymes for S phase, DNA replication enzymes. So E2F is key here, and it's one of our delayed early genes. But when E2F is made, it's not going to be immediately active, okay? So E2F, and we'll learn in the next class probably that if E2F is active and out of control, the cell's going to die. It's going to go into apoptosis. That's not important now. We'll get to that later. But at any rate, E2F is made as a delayed early genes, and it's immediately grabbed by RB. RB is named for the cancer that it was found in, retinoblastoma. So this is a really important tumor suppressor. It's mutated in a lot of cancers because it's going to block the cell cycle right here. So this is what's going to cause the cell to be blocked at that restriction point, RB bound to E2F. E2F is already on the DNA, and we'll see later it's already got a transcriptional activator with it, but this is blocked. Now, I said before, and it did not lie, that one of the keys to getting through this is going to be cyclin D. So another one of our delayed early genes is cyclin D. So cyclin D and CDK4 and 6 are going to get us up to that restriction point, and we'll see in a minute. CDK2 and cyclin E will get us through it. But at any point, any rate, our cyclins and CDKs are going to phosphorylate RB. And when RB is phosphorylated, it releases E2F, and then boom, we get our S phase enzymes, right? Things like DNA polymerase, helicase, topoisomerase, telomerase, all those enzymes we need to replicate. How you doing? Okay. All right, so that's kind of like the simple way of looking at it. Your DEGs come up, which are E2F and cyclin CDK. E2F gets grabbed. When there's enough cyclin CDK, you phosphorylate RB, and boom, you're through restriction point. But it's a lot more complicated than that. And once we get through this restriction point, we've got all kinds of cyclins and CDKs that are going to be involved in keeping this cell going forward in the cell cycle. So it's important that each of these cyclins and CDKs are going to turn on specific processes in the cell. And once that process is started, the cyclin will get degraded, and that'll shut off. A new cyclin comes in, carry the cell further, and then a new cyclin CDK carrying it further, new cyclin CDK carrying it further. And that process keeps you moving forward in the cell cycle. Now, what actually is going on as we go through this, we call this the cell cycle clock. Cyclin D, which is controlled by its transcription, right? This is being controlled by our growth factor. Cyclin D and CDK4 is going to get us up to that restriction point. Then cyclin E and CDK2 will get us through the restriction point and into S phase. Once we get into S phase, cyclin E is going to be degraded. Okay, it's actually going to activate its own degradation, just like MPF does. When cyclin E is gone, then cyclin A is going to be coming up, and it is then going to bind to CDK2. So you can see we've got the same CDK, we've just switched cyclins here. Now, the cyclin A CDK2 is going to get you most of the way through S phase, but then CDK2 is going to drop off, and cyclin A is going to pick up CDC2, which is also CDK1. We've seen this guy before. So CDK2 and cyclin A then is going to get you through S phase and through G2 right up to that G2M transition point, and then cyclin A gets degraded. And then we're going to wait for our MPF to get activated, And this is the transition we talked about on Monday. So that gets us into mitosis, and then cyclin D gets degraded, and we're back at G1. So it's kind of a complicated dance, but I think it's beautiful. I really enjoy this. So I really enjoy this complicated stuff. But anyway, here's a different way of looking at it. Exact same thing, looking at a different way. Here, I drew this figure because this is the way I think about it. Here are your CDKs, and here are your cyclins. So end of G1, CDKs 4 and 6 are going to be activated by cyclin D. Okay, that's pretty normal. And now CDK2 picks up with cyclin E, which gets you into S phase. And then cyclin A is on CDK2, gets you halfway through. Now CDC2 with cyclin A up to here. And now cyclin B with CDC2 will get you through there. So same thing as the last figure, just a different way of looking at it. Okay, so what's keeping us going forward in this cycle, as I said, is the degradation of humanity. No, the degradation of the cyclins. So we saw this before, G2M, you activate APC, and you get ubiquitin-mediated degradation of cyclin B, right? And now that's going to stay low until we need it again back here. Now cyclin D, let's come back to cyclin D. Cyclin E comes up right at the restriction point. You get into S phase, gets ubiquitinated and degraded. Cyclin A picks up on that same CDK, peaks, then it gets ubiquitinated and degraded. And we're back to cyclin B and its process, okay? So you have to keep moving in the cell cycle because the cyclins needed for the previous steps are gone now. They've been degraded. The only thing that isn't controlled by ubiquitin is cyclin D. Cyclin D is only going to be controlled by transcription factors, MAP kinase, activating your mediate early genes. Now, once you get into the restriction point, this is pretty important. Once you get into the restriction point, the cell's no longer responsive to growth factors. So at that point, cyclin D is going to drop off, but not from ubiquitination, but from it's just not being transcribed anymore. And so just natural degradation of the protein that goes away. So that's kind of an important thing to remember, something that someone might ask on a test someday. All three of these are controlled by ubiquitination. Cyclin D is not, it's only controlled by its transcription, which is tied to growth factors. And once those you get into restriction point, and that's kind of another important point. Once the cell is only responsive to growth factors up until you get through the restriction point. After that, growth factors no longer have any effect on the cell until it gets back into G1. Okay. Does that make sense? Okay. Uh-oh. Princeton, New Jersey. I think it's that Nigerian prince I've been talking to.
He goes to Rutgers?
Goes to Rutgers, yes. He's exiled at Rutgers until he gets his throne back. I'm going to help him. Okay, another big part of the story. Now, in your book, in the book that I told you to buy or to use, it doesn't really talk that much about these cyclins, about these CDK inhibitors. It just says that they block the cell cycle, which is true. They're going to block CDKs, that's going to block the cell cycle. But the story's more complicated than that, as we'll see in a minute. So anyway, you've got two different classes of CDK inhibitors. You've got your INK class and your KIP class, KIP or CIP. And the INK class, the most important ones are P15, P16. These are going to block cyclin D, CDK4/6. So that's going to stop the cell at the restriction point. And then P15 especially, the reason I say that's important is one of the growth factors we talked about in the last test was TGF-beta. TGF-beta in some cells activates growth, but mostly it's blocking cell division. And it's doing that by upregulating P15. So when P15 comes up because of TGF beta, you don't get to the restriction point. You just blocked in G1. But anyway, these guys, less so P57, it's the same thing, but P27 and P21, these are going to block everywhere else in the cell cycle. It's going to block your transition into S phase, block you in S phase, block the G2 to M. So these are really blocking most of the cell cycle. These are only blocking before that restriction point. And these are even more complicated because these are going to stimulate this. What? CDK inhibitor stimulating? Yes, indeed. Okay. So P21, when it binds to CDK 4/6 and cyclin D, it's going to activate it. Same thing with P27. We're activating these. And that's going to help the cell get up to a restriction point. But to get through that restriction point, you're going to need this, cyclin E, CDK2, and your P21 and P27 are going to block that. So the way you want to think about G1 is you're going to start G1 with a lot of P21 and P27 around. And that's going to stop you at the restriction point by blocking this. But it's going to help you get to the restriction point by stimulating this. So see these CYP and KIP CDK inhibitors are helping you get up to the restriction point, but then blocking it there. So how do you get through that restriction point? I'm glad you asked. So here we are early in G1, we've got a lot of CDK inhibitors. As you start making your DEGs, right, cyclin D, and your kinases, the CDK inhibitors are going to block your cyclin E CDK2s. And we're stimulating the Ds and CDKs 4/6. As you get later into G1, now you've got pretty much all of the CDK inhibitors binding to something, either this or this. And now as you get further into G1, you get even more cyclin D and more CDK4 and 6. And so now there's, the CDK inhibitors are going to bind to those instead. And now you've released your cyclin E CDK2. So it's kind of a numbers game here, where at the beginning, the CDK inhibitors are blocking E CDK2 until you get more cyclin D being made to give you more of this complex, and then that pulls those CDK inhibitors off, and now we've released cyclin E CDK2. And that's part of our transition. Right now we've got this guy ready. And what is that doing? We're affecting RB. Now to get back to this, we talked about it. I said this is a simple way of looking at it. It's going to be more complicated. Here, back here I said we're phosphorylating RB, and that's going to get us through the cycle. Well, here we're going to see how it's a little more complicated than that. So at the beginning, when we've got cyclin D and CDK4/6 at the beginning of G1, we hypophosphorylate RB, and that's really important. The unphosphorylated, when you pass out of mitosis into G1, that RB will be totally unphosphorylated. CDK4 and 6 then will hypophosphorylate RB, and when it's hypophosphorylated, it will bind E2F. I should have that in here. That would look cool. So there's E2F. Okay. So when it's hypophosphorylated, it's binding E2F. Then when you go through this whole process with the CDK inhibitors, and this guy gets released, cyclin E CDK2 is going to hyperphosphorylate E2F, and that's going to then release E2F so it can go on and do the transcriptions. I'm a little bit obsessive about my slides. You know, it takes me about, I would say, 7 hours to put together 1 hour of lecture, even though I've done these lectures many, many times, because my slides have to be perfect. Have to be And I think in a future version, you're gonna see, oops, a different color for the C2F because I prefer a different color. Okay. So bottom line, you've got your CDK inhibitors blocking E2F, blocking cyclin E CDK2 until the CDK cyclin D CDK4/6 outnumbers it. Then we activate this. And when we do, we hyperphosphorylate RB and release E2, and then we go through the restriction point.
Is the reason it unbinds from the cyclin E CDK2 and binds to the cyclin D? Is it because there's a higher affinity for P to the cyclin Ds, or is it just because?
That's a good question. So the question, I don't know if you can hear the questions from the crowd over here. So he said, is there a higher affinity of the CDK inhibitors for this complex to this complex? I don't know. That's a good question. As a pharmacologist, I like to think everything's about affinity. But I don't really know. It could just be a numbers thing, that this could even be higher affinity than this, but you got more of this, so you all compete it. Yeah, I don't really know, but that's an interesting question. I'll put that on the exam as one of my essay questions. See what you guys think. Okay, so that's getting us through our point. Now, like with the G2 to M transition point, we want to go through this quick, right? You don't want to slowly go into S phase. You want this to happen really fast. So we've got two positive feedback loops that are going to make this a rapid transition. One of those positive feedback loops is this. So we've got unphosphorylated RB, hypophosphorylated RB. Now it's primed to be hyperphosphorylated. This has to happen first. So now we've got our blocked E2F, and then this hyperphosphorylates it, releases E2F. E2F is a transcription factor. One of the genes it's going to turn on is cyclin E. Cyclin E gets made, activates more CDK2, more hyperphosphorylation. So we've got this positive feedback loop here between E2F and CDK2. The other positive feedback loop then is this, which is you've got your active cyclin E CDK2. It's going to phosphorylate P27, and that's going to make it come off, which is going to activate more cyclin E CDK2. And it's also going to make this a target for CUL1. Oops. CUL1 is another E3 ubiquitin ligase. So CUL1 then is going to cause degradation of P27. So then here's your positive feedback loop. This causes release of more of this, which then causes more release of this. So again, positive feedback loop, you get activation really quick. Yeah. How's that? Good. Okay. All right. So I think
you're right before though, where it's the cyclin D. Is that technically the one that's stimulated or unstimulated? That's phosphorylating, hypophosphorylating.
This guy here?
No, the one right above it, the cyclin D and CDK4. Is that the stimulated version or the unstimulated version that's causing the hypo?
That's a good question, because you would expect it to have a
right.
Yeah, a CDK inhibitor here. Yeah, I don't know. From here, it looks like whether it's stimulated or not is going to be able to hypophosphorylate. I don't know. That's a good question. Never noticed that. One thing though, to keep straight in your head as you get into the next exam: hypophosphorylated versus hyperphosphorylated. Hypo is priming it. Hyper is getting you through that restriction point. And then RB is going to be hyperphosphorylated in the whole rest of the cycle until you get to the end of mitosis, and then you get activation of a phosphatase that's going to totally unphosphorylate it. And I think we'll talk about that in the next class. I'm not sure. That bomb. Here's another thing. I'm mainly throwing this out here because we talked about this a little bit in the last class. It's kind of a general mechanism of controlling chromatin availability. So we saw this in the last exam with lipophilic hormones. If you remember, some lipophilic hormone receptors are bound to the DNA, but they bring with them the histone deacetylase. And that's going to deacetylate the histones and cause them to condense the chromatin up. So here is hypophosphorylated RB. It's bound to E2F, and E2F's co-activator is DP12. So these are bound to the DNA, and RB is bound to it to inhibit it, and it's also bringing in histone deacetylase. So we're condensing the chromosome up here. And then when you hyperphosphorylate and relieve that stress, histone acetylase comes in, acetylating the histones and opening up the DNA here. So I like this because it's a callback to our lipophilic hormone receptor mechanism. And it, brings into play this whole idea of like levels of regulation of genes. We always think of genes being regulated by transcription factors, which is true, but also they're also regulated by how tightly wound or how loose they are, because when they're bound up with histones, the transcription factors can't get to them anyway. So, or the polymerases or whatever. So anyway, this is another mechanism of regulation of the genes. And then I think, oh no, I've got plenty more. Good. Okay, I was afraid of running out of notes, but I'm like, oh, this whole semester has been like trying to stay one class ahead of you guys. So anyway, this is an ugly figure that I don't want you to memorize. The only thing I want to point out here is these are a lot of the pathways we've already talked about. And all of these pathways lead to cyclin D. So this is our RAS pathway that we just talked about. HER-NU, I don't really know that pathway. Here's our JAK-STAT pathway, Hedgehog, NF-kappa B, which is another one that is pretty important in a lot of things. Here's our WNT, here's our PI3 kinase, beta-catenin here. Everything leads to cyclin D. Cyclin D is one of our DEGs, delayed early genes, and that is the key to getting us up to and then through the restriction point. All right.
This is kind of going ahead.
Uh-huh.
I don't know if it's something that we'll cover later, like HeLa cells and where in this like whole cycle is mutated and what's changed.
What's changed in the cells? Yeah, we're going to talk about that toward after we get through how the cell divides. Then we'll talk about how when you mutate different things, it stops the control over the division. Yeah. And I'm going to try and stay ahead of you on that, so I know more than you when we get there. HeLa cells. I remember HeLa cells from when I was in grad school. Some people were working with HeLa cells a long time ago. Okay, P53. Should we do P53 or do you want to take a break? Spring break. Let's do P53. So you've probably all heard P53 before. We call it the guardian of the genome because it's kind of central in terms of protecting the cell from dividing with damaged DNA, because that's what's going to cause the cell to become cancerous. When you start mutating some of these pro-growth regulators like growth factors, kinases, all these things, when you lose control over those, the cell can't control the cell division. And that's all about DNA mutations. So P53 is central to controlling the cell's response to DNA damage. And the things that are going to turn this on are like hypoxia, right? Hypoxia can cause a lot of DNA damage in general, oncogenic signaling, oxidative stress, hormones, all kinds of things can stimulate P53 to come up. And when P53 comes up, there are two different possible things, actually three here. One is the first thing that happens is the cell arrests and it stops, and then it corrects that DNA damage. But if there's too much DNA damage, it will go into apoptosis and die. So let's talk about that. So here, This shows you how P53 protects cells. So these are mice that are transgenic, so you can control how many P53 alleles there are. So a normal mouse can live, 500 days or whatever a normal mouse lives. You knock out one of those P53s and you're decreasing their lifespan. Okay, this is 70% survival at 500 days. If it doesn't have any P53, boom, the lifespan drops off precipitously. And these deaths are due to cancer. And that's why P53 is mutated in like 50% of all human cancers. Because if you don't have a P53, it doesn't cause cancer, but it allows cancer. Because it allows cells to keep dividing with DNA damage.
So you pretty much have cancer on day one if both of your P53s are knocked out.
Yeah, there's a thing they talk about in the book called Li-Fraumeni syndrome or something like that, where you inherit two null P53s, and they have all kinds of cancers all through childhood in a very short lifespan.
It just goes to show you how many mutations this thing is stopping. Because if it's that frequent that your
That's right.
50% survival rate is 100 days.
Exactly. Good point. You can tell how much DNA mutation there is, just repeating for the crowd.
Yeah.
You can tell how much DNA mutation there is on a regular basis when you can't control for it. It's just out of control. So there's an awful lot of this is a very active process. Again, here are the things that activate this. What we're most interested in is UV radiation, ionizing radiation. These are going to directly mutate bases. Oncogenic signaling. We'll see this later where if E2F is like extremely high, like you way overstimulated E2F, that will affect P53 and cause the cell to go into apoptosis. Hypoxia, blockage of transcription. These three in the middle are the ones we mostly are going to be talking about. P53 is a tetramer, so they draw it this way. When P53 increases in the cell, you get cell cycle arrest, so it stops dividing, and then you get upregulation of DNA repair enzymes to fix it. You're also blocking angiogenesis, and if the DNA damage is too great, you go into apoptosis. And I always like to tell the story of when I was a stupid college student. Senior year in college, I went down to Florida. Being from Pennsylvania, I had never really seen the sun. I thought I had seen the sun, didn't realize that the sun is very different in Florida. First day there, I laid out in the sun for like 2 hours. No sunscreen. This is 1985. Sunscreens for women. So I spent the next 3 days in the hotel room. Blisters on my forehead, blisters on my chest, totally burned. And a couple days later, the skin started coming off in big shards. Okay, so basically UV radiation causes thymine dimers, and so I got massive DNA damage in my dermis or epidermis, and like all those cells went into apoptosis. So I'm a big fan of sunscreen these days, and you can see from my face all the sun damage over the years because I was just stupid. But anyway, my children use sunscreen. Okay, so here we are. This is another experiment. Looking at P53 and UV damage. So here, what they did was they took a whole bunch of cells and they zapped them with enough UV to kill all the cells. So here's time 0. At time 0, P53 is really low. And then they hit it with UV. 8 hours later, P53 is really high. And then 24 hours later, we get production of this CDK P21 to block the cell cycle, and P53 is really high. So after UV damage, you're getting upregulation of P53 and then upregulation of this CDK inhibitor to block cell cycle. Over here, we're looking at cell survival. Here with 2 good P53 versions, cells die pretty quickly. And that's P53 mediated apoptosis. With only 1 version, you're still getting cell death. Okay, so these cells are still dying. No P53, they don't mind. They're just going to keep going. Okay, unless you've knocked out some important genes with that UV radiation, they're just going to keep dividing. So very important in protecting against DNA damage. And this is how this works. Now, your book goes into tremendous detail here with ATM, ATR, and checkpoint kinases. And I think after spring break, we're going to get into that as well. Right now, all we want to do is just assume this part of the figure and then move on to this part of the figure. So if you've got DNA damage, it could be double-strand breaks. Double-strand breaks activate the enzyme ATM. Could be single-strand breaks, and that activates ATR. A lot of other type of damage will activate these checkpoint kinases as well. So once you activate these, you activate these, which is like check 1 and check 2. And these are going to phosphorylate P53. Now, in the absence of DNA damage, we've got this bad boy here, MDM2. So MDM2 is another E3 ubiquitin ligase. It's going to hold on to P53, add change of ubiquitin, and then it's degraded. So just like we saw with beta-catenin, we're constantly breaking down P53. Right? So it's constantly being made, constantly being broken down. So its concentration is controlled by degradation. And MDM2 is the enzyme that's going to be ubiquitinating it and causing its degradation. Now, when you've got DNA damage, you activate these checkpoint kinases, they phosphorylate P53, and then it's protected. It's no longer a substrate for MDM2. And two different paths we can go on this, depending on how much DNA damage there is, depending on whether you use sunscreen or you didn't. Okay, if you've got a little bit of DNA damage, you're going to activate P21, which is going to block your cell cycle, right? Cyclin E, CDK2 is going to be blocked. And then you're not activating RB, cell cycle arrest. You're also activating DNA repair enzymes to fix that damage. If there's a tremendous amount of DNA damage, then P53 will activate PUMA, P53 upregulated modulator of apoptosis. This is a pro-apoptotic BCL2. So we haven't talked about BCL2s. We'll get to them after spring break. But this is then going to inhibit the anti-apoptotic BCL2, which then allows apoptosis to happen. So a little bit of damage, we're going to block cell cycle and fix the damage. A lot of damage, we're going to activate apoptosis and let the cell die. Okay, so that's the bottom line with P52. Always being broken down until there's DNA damage. And then depending on the amount of damage, we're either going to fix it or commit suicide. And any question about that before we move on? Okay, just let's see how much more I can talk about. Just a couple more things to talk about. So the interesting thing about P53 and MDM2 is they kind of regulate each other. Here's following concentration of MDM2 and P53 and how they follow each other. So what's going on here is MDM2 is going to ubiquitinate P53, right, and brings it down. Now, when MDM2 decreases, you get less ubiquitination of P53, so it increases. P53 then causes the transcription of MDM2. So when P53 gets high, you get more MDM2. It then ubiquitinates P53 and it comes down. Now, P53 is low. It's no longer transcribing MDM2. MDM2 comes down. P53 goes up. And then it causes the transcription of MDM2. MDM2 comes back up. P53 goes down. So it's kind of interesting how they interplay that way because MDM2 is causing the degradation of P53. P53 is inducing MDM2. They're interrelated there. I'm not sure if that's all that interesting in terms of how the cell cycle is controlled. Here's something that is really, really interesting. So earlier we talked about interesting and important. I should make that distinction. I find a lot of things interesting that aren't important. Here we talked about totally losing P53, right? But there are worse things than totally losing P53. And that is if you mutate just one of the two alleles. So P53 acts as a tetramer. So there are point mutations point mutations. I keep thinking about beer. There are point mutations that will inactivate P53, but still let it go into a complex with the normal. So here we're talking about a situation where you've got two alleles with P53. One's normal, one's got a point mutation that makes it inactive. Now, the cell's making protein from both of those genes, so you get both the normal and the mutated made at the same time. And they're randomly binding to each other. So just based on statistics, if you have at least one of the units of the tetramer with this point mutation in, the whole tetramer is inactive. So, and just based on statistics, if you've got a tetramer, then 1 out of 16 will have 4 mutateds, 1 out of 16 will have all normals, and all the other ones have a combination of mutated normals. Now, everything with at least 1 mutated is inactive, which means only 1 out of 16 of your tetramers is active. So this is what we call a dominant negative. It's mutated and it's shutting off the activity of the good gene. Okay?
And this is different from having one P53 knocked out as before.
Exactly. So before we're like, where is it? Shoot. I've got a 13-year-old in my house and my language has gotten so bad. So before we're saying, here's one normal gene and one totally knocked out gene. Maybe it's not even transcribed anymore. And it's almost normal, right? Because you have that one normal P53 there. So here what we're saying is you got both genes being made, but one's got a point mutation and that knocks out the activity of the other one, or most of it anyway.
Right. So if the other one's not being transcribed, they're all good. You just don't have enough of the tetramers.
Most of the tetramers are inactive because they've got at least one subunit that's got a point mutation.
No, of the heterozygote where you have a
Oh,
P53 knocked out.
okay, yeah. So now you're making good P53 from the one gene that's good, and they, tetramerize up, and they're all active. They're all fine.
You just don't have enough of them.
You just don't have enough of it. You've got less because you only have one gene. Yeah. Okay. So this is what this is showing here. So if you've got a total deletion, P53, so you've got one wild type gene and one null gene, you're almost normal. But if you've got a wild type gene and a point mutation, you have almost no P53 function. This is just a
Which is just as bad as having both knockouts.
Right. Just as bad as having them both knocked out. And this is kind of neat. What I like about this is the cell sorter aspect to it. So you guys know what a cell sorter is? Did you ever hear of this? It's like you've got a plate full of cells and you want to determine the distinction between different cells. So you lift them off the plate and you put them in a solution and you run them through this instrument. And it's basically a capillary tube that makes the solution goes through and cells go through one at a time. And as they're going through, you've got a light source that's measuring something in those cells. And then maybe you're measuring the amount of DNA, right? And so you can tell what cells are in G1 because they've got a normal amount of DNA, what cells are in G2 because they got twice as much DNA, and you can also tell cell size. So what we're looking at here is cell size. So here in the top layer, you can see all the cells are pretty much the same size, right? You've got a bell-shaped curve right in the middle. Now down here, you've got a whole bunch of small cells, which basically means apoptosis. These cells are breaking apart into little apoptotic bodies. So this is, then I probably shouldn't be telling you about this because this is about the next slide. What this is telling is something that I alluded to before was if you've got too much E2F, the cell's going to go into apoptosis. So here we've got a chimera between the estrogen receptor and E2F. So we put this into some cells, and now without estrogen, OHT is estrogen basically. Without estrogen, all the cells are normal. We add estrogen, we activate E2F, they all go into apoptosis. So I'm going to leave you with that thought. We'll talk about it in the next class, but this is how, if a cell is overstimulated, so it's oncogenic, it's gonna have a really high level of HOF. Normally that cell will go into apoptosis. So it's kind of like a fail-safe to protect the cells. And oh, this is a gorgeous mechanism. This is why. So I'm not gonna talk about that today. I just want to keep you on the edge of your seats all the way through spring break. Oh, 12:15, I guess we're done. Any questions? Anything I want to go back over? How about the crowd? How are we doing back here? Let me stop sharing. Any questions from the peanut gallery? No questions? All right, so unfortunately, I'm not going to see you guys again for another week. Wow. Oh, it's going to be a good week for me though, because I get a chance to catch up with Plattsburgh and get ahead of you guys. And then when we get back, it'll be some more stellar.
I think you have a chat in the.
No chat? I think Isabella just said thanks. No questions, thank you. Okay. I should just open that chat when we're here anyway. Okay, all right, I'm gonna say goodbye. And I'll see you all guys, at the other side of spring break.